Please give a warm welcome to Dr. Leila Strickland, CEO and co-founder at BIOMILQ. Hello.
Thank you.
Hello, Leila. The stage is yours.
Thank you. Fantastic. Hi. Thanks, everybody. Hope everybody's having a great time at the conference. My name's Leila, and I can't wait to tell you guys about my company, BIOMILQ, where we're developing mammary biotechnology to produce bioauthentic human milk products. Let's go ahead and get into it. Have you ever really thought about breast milk? Like, not in a weird way, but in a nerdy way? Like, if you're a parent, you probably thought about it a lot in the early days, weeks, and months of your baby's life. It's a substance that is, harvested from the mother in order to build the body of the infant. It's pretty remarkable. At the molecular level, human milk is filled with a unique profile of proteins, lipids, carbohydrates in the form of human milk oligosaccharides, or HMOs, which we just heard a little bit about. And they're exquisitely tuned through our 200 million year history of evolution in order to specifically advantage human physiology, all aspects of human physiology. But there's a real supply and demand problem with human milk, when even its natural consumers, our babies, don't get enough of it. And without a technology to produce it, the rest of us don't even think about what human milk components could do for our own bodies as we proceed through our lives and experience inevitably injury, illness, aging. So at BIOMILQ, what we're working on is a mammary biotechnology to produce the bioauthentic versions of human milk components to benefit all types of consumers from birth and beyond across the life cycle. Of course, there are a range of approaches that can be taken to try to partially recreate some of the beneficial aspects of human milk. Infant formulas today are largely based on bovine milk, which can safely be processed and recombined into compositions that better reflect the composition of human milk. But there are real limitations with both the structures and the proportional relationships that are present in bovine milk and just cannot be reformulated truly into an equivalent of a human milk product. And increasingly, we feel urgently that it's important to move away from a reliance on animal agriculture to produce these types of products. And so we see the emergence of new technologies, alternative protein, microbial biomanufacturing, or precision fermentation, in order to develop a handful of components found in human milk. These technologies tend to be scalable. That's the good thing. But they're relying on microorganisms that lack the biological bells and whistles that are required to produce really the most functional forms of these types of components and ingredients. And furthermore, for every single molecule produced in a microbial biomanufacturing environment or precision fermentation, you need an entire bioprocess. And so this makes it really difficult to imagine approaching a substance as complex as human milk, which contains hundreds, if not thousands, of unique components. So this is where BIOMILQ comes into play. At BIOMILQ, we understand that the beneficial aspects of human milk are a direct consequence of the biological environment in which it is produced. The human mammary epithelial cell is the only cell type on Earth that can really, truly approach the functional complexity of human milk. And my team at BIOMILQ is full of cell biologists, genetic engineers, and bioprocess developers who are working on culturing human mammary epithelial cells that we isolated from breast milk and have now converted into a production system to approach the certain components of human milk, which I'll tell you about a few of today. So we're really working on two core programs at BIOMILQ right now, and I'm going to tell you specifically about our osteopontin program. This is a human milk protein. But before I get into it, I want to talk a little bit about how we choose a program or how we design a program at BIOMILQ. How do we decide in this kaleidoscope of molecules what should we work on? Milk is so complex. There's so many things in it you could go after. And so when we look at it, we really want to think about molecules that are inaccessible with current technology, don't have another method of production, can't really be made in their most active functional forms using a microbial fermentation, or are not present at high levels in bovine milk, or not in the same structure and function. We want to look at molecules that have a history of literature backing the idea that they have known and potent benefits for human health. You should only need a few hundred milligrams, perhaps, of these molecules every day instead of grams and grams of them. That's really important for the type of technology that we're developing. And then also, we want to see some kind of a signal that there's perhaps an appetite for these molecules in the market, so that as we start to roll out our first products coming from our platform, we know that there's going to be a marketplace to receive them. So osteopontin, this human milk protein that you may or may not have heard of yet, but will certainly be hearing more about in the future, checks all of these boxes. It is a protein with a complex structure that cannot really be recreated using other technologies, and the human version is different than the bovine version. It's been studied for many, many years and is well known to have a lot of different important biological effects in our bodies. And we're beginning to see it emerge as an ingredient in infant formulas today in Europe and Asia. So a little bit about osteopontin. This is, as I mentioned, a protein where we know that there are benefits associated with this protein throughout the whole human body. So as the name implies, osteopontin is involved in bone mineralization and therefore has an important role to play in skeletal development. That suggests that it could also potentially be valuable for people who are recovering from a bone injury. And then beyond its role in bone regulation and bone mineralization, osteopontin has been implicated in immune modulation, the inflammatory response. It apparently modulates the microbiome. It's got roles to play in wound repair and even brain development. So how does one protein do so much? To answer that question, you have to think a little bit about its biochemistry and its structure and its function. So on the right, I'm showing you a diagram, just looks like a blue ribbon that shows the length of the osteopontin protein. And along the length of that protein, it's decorated with these little blue balls and these green forks, those calcium ions shown in gold. And these are called post-translational modifications. These are specific changes to the protein structure that are made in the cell after this protein has been synthesized. The changes in these structure impact every aspect of how this protein interacts with the other things it comes into contact with as it moves throughout your body. And so they have a very important role to play in its function and how it behaves. The phosphorylation of osteopontin is particularly important as it's high up in the pathway that ultimately results in the most functional forms of this protein that occur in breast milk. Osteopontin is made in almost every cell type in the body, but the breast milk form of the protein is specifically different. And it's different because of the phosphorylation profile that is applied along the length of the structure. So at BIOMILQ, I told you about how we're working with human mammary epithelial cells. And our hypothesis was that only human mammary epithelial cells would be capable of really recreating this structure, and that would begin with generating the correct phosphorylation profile. So the slide I'm showing now is a bit of data from my lab showing that exactly that is what we saw. We obtained a human osteopontin protein that had been produced in a bacterial system, as well as in two different versions of a mammalian system that's very commonly used in biomanufacturing. And then we just simply counted the number of phosphorylations added to this protein during its synthesis. And you can see in the BIOMILQ bar, the purple bar at the bottom, we achieved the highest number of phosphorylations. We got 21 in this particular experiment. And that was great news and exactly what we hope to see, because breast milk osteopontin will on average contain 20 to 25 different phosphorylations. So this was a signal for us to continue to develop this program, and we now see this as an important market opportunity for BIOMILQ, along with other programs in our pipeline. The osteopontin market is expected to reach around $800 million by 2032, with 13.5% growth year over year. And that's driven by a growing demand for functional ingredients in infant formula and early life nutrition, as we heard a bit about just a moment ago. And we see the benefits of these proteins and products being important throughout the entire life cycle. I'll just close by representing that, I have we're a venture-backed early-stage company. We're a Series A stage company. I have a team of 25 brilliant people working on this in our lab in North Carolina. Our work has culminated in 5 different patents that are moving their way across the globe. And the portfolio protects the work we've done in cell line development, starting with our 60 primary cell lines, our 6 immortalized cell lines, and the 2 programs, which are including osteopontin, which I've just told you about, and human milk oligosaccharides, which we'll be discussing more in the future. So I think I have run out of time, but we're looking to partner with other companies who are interested in bringing the future of human health through human milk ingredients. And if any of this is of interest to you, you can find myself and my team at our booth next door. That's all. Thank you.